introduction.
Yang-Nan Gu from the University of California in Berkeley in the United States. And he's going to talk about his current progress on understanding the plant nuclear envelope membrane. So, Dr. Gu, please.
Well, thanks, Manja, for the introduction. So it is really a great honor to be one of the finalists. I really want to thank the work committee for giving me this opportunity to share our work. So our laboratory is actually interested in understanding the plant nuclear envelope. We know that nuclear envelope or nuclear membrane is a hallmark of eukaryotic life. It isolates the genetic material from the rest of the cells and physically separate the transcription and translation in two different compartments, which enables the evolution of numerous regulatory mechanisms to ensure the success of eukaryotic life. So emerging evidence have indicated that this double-layer nuclear membrane is not only a physical barrier, but also a versatile communication interface and organization platform. These functions are actually achieved through selectively enriched a group of proteins that are particularly localized to the nuclear envelope that function in many of the essential nuclear processes. For example, we know the nuclear pore, right? So on top of the nuclear pore actually sits the largest micromolecule protein complex called the nuclear pore complex, which is composed by over 1,000 proteins of 40 different kinds. They assemble into a large channel-like structures which can mediate selective and bidirectional transport of many macromolecules, including RNAs and proteins. Another example is this kind of protein complex called the link complex for linkers of the nuclear skeleton and cytoskeleton. So this complex can function to transmit the mechanical forces from the cytosol or even outside of the cells into changes in the nuclear skeleton and chromatin structure changes in order to initiate differential gene expression in response to mechanical forces. So these are just two examples. There are about 300 to 400 nuclear envelope resident or associated proteins found in both human and yeast. Well, they play important roles, and mutation of those nuclear envelope proteins can cause serious diseases. One of the most famous one is called progeria. It's an early aging disease. Patients usually cannot survive until they're teenagers, and they have very serious deformed nuclear morphology compared to normal people. And this is caused by a mutation in the nuclear skeleton proteins called lamin. Okay, unfortunately, this is not the only examples. Actually, mutations in many of the nuclear envelope genes are tightly associated with a series of tissue-specific diseases in humans, ranging from muscle dystrophies to neuron degenerations or cardiovascular diseases and immune deficiency. And those diseases are actually collectively known as the nuclear envelopathies. And there's many of them, and the list is actually continues to expand. Well, we are interested in understanding the nuclear envelope in plants. So there's two interesting facts I need to point out for plant nuclear envelope. The first is that although there's hundreds of them identified in human and yeast nuclear envelope proteins, but this nuclear envelope protein number identified in plant is surprisingly low, almost a order of magnitude less than what is found in human and yeast. So that indicated we really know very little about the information of what is the composition of the nuclear envelope in plants. So the second interesting fact is that except for the nuclear pore complex and the link complex that I talked about before, most of the plant nuclear envelope actually have little or no sequence homology with their functional counterparts in yeast or animals. So for examples, we know that nuclear skeletons in humans are composed by those lamin proteins. Well, actually plants do not encode any of the lamin proteins, but they encode a gene that encode many similar structure of long helical domains that are crown proteins. They compose the plant nuclear skeletons, and they have no sequence homology with lamin at all. And also, not only that, plant don't encode any of those key essential components of nuclear envelopes in animals, for example, amberine proteins, laminin receptors, or LAB2 proteins, or LAM domain containing proteins. Plant don't have them at all. So that raises an interesting question. It seems like the plant nuclear envelope really has independently, or at least partially independently evolved from the rest of the eukaryotic cells. So what is the component or protein composition of the nuclear envelope in plant? And what are their functions and how they are evolved? Why they evolve differently from the rest of the eukaryotic life is actually open questions at this moment. So the reason that got me into this particular area is because during my postdoc training, I actually found one particular plant nuclear membrane proteins that plays very, very important roles in regulating the plant immune responses. This protein is called CPR5. It encodes a multiple transmembrane protein without other identifiable domains. Loss of function of CPR5 cause serious autoimmune disease in Arabidopsis, including highly expressed defense gene and spontaneous programmed cell deaths in the absence of any pathogens. Well, to make a long story short, through a series of genetic and molecular and biochemical characterization, I defined that the CPR5s actually compose a bona fide component in the nuclear pore complex in plants. It forms this oligomeric form, which is very important to maintain the functional and structure of the nuclear pore complex under normal conditions. However, in the presence of pathogen infections, especially those that carry effector proteins that can activate NLR type of immune resistance, the CPR5 actually undergoes a monomerization process. And this process actually changes the nuclear pore complex conformation and results in unspecific accumulation and transport of a series of immune and stress signaling cargoes in the nucleus, which eventually activates the transcription amplification of immune responses and result in a programmed cell death. So in this study, we basically established a concept that the nuclear pore complex is actually not a static channel for just the transport. Instead, it is a flexible environment that are able to undergo dynamic changes in response to environments, and in this case, pathogen infections. And these changes actually can regulate the transport capacity of MPC, and in order to facilitate the environmental mediated responses. Well, this idea is actually very well supported by a series of recent publications which reveal a near atomic structure of the nuclear pore complex in humans. These findings actually reveal that the nuclear pore complex can undergo this dilation and constriction processes. And this process actually can dramatically change the nuclear pore complex central channel diameters, which potentially affect the transport capacity and the selectivity of the nuclear pore complex, which is very consistent with the model that was proposed in plant a few years ago. So study of the CPR5 protein actually got me very interested in the nuclear pore complex in plant, although it has been demonstrated to be very conserved among eukaryotic life, but plant apparently has its own component of the nuclear pore complex, its own regulatory mechanism, because CPR5 is a plant-specific gene that doesn't exist in animals or yeast. The study also made me realize that how poorly we have understand entire nuclear envelope structures and compositions. So for that, when I established my own lab, one of the major first goal is to trying to understand and profile the protein composition of the nuclear envelope in plant. So that sounds like at the first glance a question that it can be easily resolved. Okay? So you can just purify the nucleus and subject it to mass spectrometry to understand its protein composition. However, this is not that easy. There's fundamental hurdles that we need to overcome. So one of the most important one is that actually the nuclear envelope, especially the outer nuclear membranes, is continuous with the endoplasmic reticulum. So whenever we purify the nucleus, there's always inevitably abundant ER proteins co-purified. There's no way to distinguish between ER and nuclear envelope. So to overcome this problem, we actually developed two different proteomic strategies. So the first one we call subtractive proteomics, in which we first purify the nucleus from Arabidopsis. And this purified nucleus, we actually know it contains a lot of contamination, membrane compartment from other organelles. But at the same time, we'll purify the total microsomal membrane. So we subject both the fractions into quantitative mass spectrometry, and in the result, we applied subtractive analysis in order to subtract anything from the total microsomal membrane from the NE fractions to retain only the NE specific fractions components. So the pros of this technology is that we actually end up with a very specific subset of proteins that seems to be very comprehensive, covering the nuclear envelope of known plant origin Arabidopsis. For example, we covered almost 90% of known nuclear pore complex and nuclear envelope proteins in Arabidopsis, which means this is a very comprehensive subgroup of proteins that if there's any bona fide nuclear envelope or nuclear pore complex component, it should be within this group. However, the cons of this method is actually during our validation of those membrane proteins, we found it actually have a very high false positives. Nearly half of the membrane proteins that we perform the localization actually localize in membrane compartment other than the nuclear membranes. So to overcome this problem, we developed the second proteomic strategies, which we call proximity labeling proteomics. So for those who are not familiar with the proximity labeling strategies, it's actually very simple. You can fuse your protein of interest with a promiscuous biotin ligase from E. coli, so that later it can biotinylate any protein that comes close to your protein of interest or directly interact with your protein of interest. And then you can just break down the cells and affinity purified all of those biotinylated proteins and subject to mass spectrometry for identification. So in our case, we can fuse a known nuclear membrane protein with those bio-IDs or biotin ligase and use the proximity labeling to identify any of those nuclear membrane resident or associated proteins. Well, to make a long story short, this method actually working surprisingly well. It shows unprecedented specificity and efficiency. And combine those two different proteomic strategies, we actually now were able to identify a large number of plant nuclear envelope transmembrane protein we call PNAT proteins. And we have validated 14 of those as bona fide nuclear envelope protein in plant. So we're not going to stop there. At this moment, we're trying to use this newly identified and validated PNAT protein and set up a new round of proximity labeling proteomics, trying to further expand our categories. So our ultimate goal is trying to establish a complete and precise categories of protein landscape in Arabidopsis, first in Arabidopsis and then in other plant species. So although we don't have a complete map at this moment, but using the newly identified nuclear membrane proteins and also a few of those previous characterized nuclear membrane proteins, and put them together, we actually already can see some of the unique features of a plant nuclear envelope compositions. For example, these are all the PNAT proteins in Arabidopsis. We can use the Arabidopsis and their homologs in different plant species, and also search for their homologs in many of the animal species and also yeast, and build up this evolutionary distance map. And in this map, the darker the color means the more sequence divergence from the Arabidopsis homologs. And from there, you can actually divide this nuclear membrane proteins into 3 different categories. So I want to draw your attention to this particular categories, which are actually only existing plants. Those categories is actually contains almost half of the all PNAT proteins we have identified at this moment. So these proteins are all plant specific. So indicating that the plant nuclear envelope are really composed by a group of proteins that are uniquely evolved or developed in plant. And also there are this second and intermediate groups. So these groups of PNAT proteins are actually very conserved among plants, but shows considerable sequence variations between plant and other species. indicating their function is probably differentiated during the evolution. So these evidence actually indicate that plant nuclear envelopes is actually evolved at least partially independent from the rest of the eukaryotic life, contains a lot of its unique compositions, and may have its unique functions. So in the next couple of slides, I want to briefly tell you our individual study of those PNAT proteins. Actually, through those studies, we actually can understand a lot of interesting information or biological process or mechanism that are either conserved among different eukaryotes or specific to plants. So the first example I want to give you is those PNAT2 proteins. So PNAT2 proteins are multiple transmembrane proteins that contain 3 homologs in Arabidopsis. Apparently, this protein is very important because the triple knockout of this PNAT2 genes shows a very severe phenotypes in Arabidopsis. So some of the seedlings do not have shoots; they only have hypercotyledons. Some of them do not have roots; they only have hypercotyls and cotyledons. And some of them don't have any shoot or root part; they only have this cotyledon part developed. So apparently, very important for development. And even more interesting for this triple mutant is that without changing the DNA contained per cell, this triple mutant actually dramatically altered the 3-dimensional organization of chromosome within the cells. This can be viewed by either Hi-C analysis or just a simple staining of the DNA with just a DAX. So to make a long story short, we actually find that the PNAT2 protein actually encoded an inner nuclear membrane transmembrane proteins. It uses its N-terminus to bind to the nuclear skeleton structure in order to anchor this protein inside the inner nuclear membrane, and it uses its C-terminus to bind and recruit a bunch of nucleosome structures. And this recruitment of a nucleosome is actually very important for determining the 3-dimensional structure of chromatin underneath the nuclear envelope. So this is actually very important not only for the 3-dimensional organization of chromatin, but also important for the gene expression regulation, as just Bob's talk has mentioned. So many of the transcription factors, epigenetic modifiers, or chromatin remodelers are actually specifically recruited at the nuclear envelope structures by interacting with those nuclear skeletons. And those are actually important to interact with the chromatin to differentially regulate the gene expression. So without PNAT2, the overall structure, hierarchical structure of chromatin got messed up, and it also affected the differential expression of genes, including growth and development, and a bunch of abiotic and biotic stress responses. So that generated a traumatic phenotype of this triple mutant. Well, the interesting thing is the PNAT2 is also a conserved protein. It has a homolog in animals, so this probably a conserved mechanism among both plant and animals. So another example I want to give you is that using those proximity labeling proteomics, we are not only able to identify many of the transmembrane components of the nuclear membrane, but we are also able to identify many protein or protein complex that are not transmembrane or resident in the nuclear envelope, but associate with the nuclear envelope and regulate nuclear membrane protein dynamics. So in this case, we use the SUM1, which is the inner nuclear membrane protein, part of the link complex, as a bait, and use the proximity labeling proteomics. We identify a bunch of proteins that are actually involved in ubiquitin-mediated proteolysis process. Well, whenever we discover this result, we actually are very surprised, and then that actually raises a very interesting question of how the nuclear membrane protein, especially those that localize in the inner nuclear membrane protein, once transported inside the nucleus, how these proteins are degraded once they reach the nuclear envelope. So why this is an interesting question is because normally membrane protein degradation are occurred in different mechanisms. For example, in the plasmamembrane localized transmembrane protein, when they need to be degraded, they undergo endocytosis into those endosome structures and go through the vesicle trafficking process and goes to the central vacuum for degradation. You also have this autophagy processes that degrade all of the other membrane-associated proteins and the organelles. And we also have this ER-associated degradation system or Golgi-associated degradation system, but ERAD or EGAD, to degrade proteins in the membranes of ER or the Golgi. However, once a membrane protein gets into the nucleus and localized inside the inner nuclear membranes, they're usually anchored there by binding to the nucleus molecules. But how those proteins are degraded, it's unlikely this inner nuclear membrane protein can use any of those mechanisms outside the nucleus to mediate their degradation. So how could malfunctioned or overaccumulated or mislocalized the protein at the inner nuclear membrane can be degraded? It's actually a question that is poorly understood in any higher eukaryotes. So why we care about that overexpression of inner nuclear membrane proteins? Because those can be highly related to the pathogenesis. For example, the progeria patients usually has a highly accumulated some proteins, and that actually can accelerate their pathogenesis. So how the inner nuclear protein is degraded? Through our analysis, mechanistically, we find that the inner nuclear membrane protein actually got polyubiquitinated once they were overaccumulated and need to be degraded. And this can be recognized by a TDP-48 trimeric complex. And this can actually use ATP and energy to drag those membrane protein out of the membranes and send it into the 26S chromosome for degradation. And we also found a series of PUX protein, which is a plant UBX domain containing proteins, can specifically regulate this process. Well, there's still gaps we need to fill in this pathway, but this is representing the first attempts in understanding what we call the inner nuclear membrane associated degradation pathway in higher eukaryotes. All right, I want to use this slide to briefly summarize a few of the interesting knowledge that we have learned from studying individual PNAT proteins about the nuclear envelope in plants. So except for CPR5, we actually find PNAT1 is also a very specific nuclear pore complex component. It's a transmembrane nucleoporins. So together with the CPR5, we actually find the nuclear pore complex actually have a significant more number of transmembrane nucleoporins compared to animals in the yeast. And these seems to be involved in different processes. For example, CPR5 is regulating the immune activation, and PNAT1 seems to regulate cell cycle progression. So that's a project we're currently pursuing. And also, not only the core scaffold of the nuclear pore complex, we recently find a particular guanine binding proteins that are very interesting called GBPL3, which specifically localized into the nuclear basket region. So this GBPL3 is very different from its homologue in human, which is known as GBP proteins, in that it actually invoked very particular C-terminal regions that are intrinsically disordered. So this intrinsic disorder region can promote phase separation out of this nuclear basket regions. And this phase separation actually can recruit a lot of transcription factors and RNA processing machinery at the same time. So basically, it's established that this nuclear basket region at the nuclear pore complex is a very active positions for transcription regulation, which is coupled by the RNA processing machinery and RNA transport machinery to facilitate the rapid transcription regulations. This work is also undergoing the review. So we also reviewed that PNAT2 is the inner nuclear membrane protein that are required for the chromatin organization underneath the nuclear envelope. And also there's the specific pathways that mediate the degradation of the inner nuclear membrane proteins to regulate their homeostasis. There are also other interesting aspects we're also currently following, for example, how the specific lipid metabolism is happening near the nuclear envelope regions by other types of PNAT proteins, which can specifically sensing and regulating the biogenesis of the steroids at the site of outer nuclear envelopes. So with that, I think I'm running out of time. I would like to acknowledge several of very talented postdocs from the labs that have done the most of the work I talked about. So Yu is, and Albo is one of the driving forces in the lab to profile the nuclear envelope categories. And Yu has published the PNAT1 and PNAT2 works. and also the GBPO3 works. OVO works on the internuclear membrane degradation process. Fifi and Min is working actually on another project about the cariformin beta protein regulating plant immune responses that I don't have time to introduce. So we were supported by NSF, USDA, NAFA, and IGI at UC Berkeley. I would like to take any questions at this moment.
Thank you, Dr. Gu. Very nice talk. Let's move on to the Q&A session. The committee members, please feel free to open up the microphone if you have questions for Dr. Gu. Thank you.
Hey, Yanan.
Hi, Sean.
Are you also working on biogenesis of those peanut proteins? I gathered probably their nuclear import might have some specific characters.
Yeah, so that's another project we're working in the lab that I don't have to talk about. So we are working on a specific family called importing cariformin beta proteins, so the importings and exportings, which can directly interact with the nucleophore complex to mediate the cargo transport. So they actually 18 numbers in this importing beta families, which supposedly can define differential regulation of different populations of cargoes in the plant cells to control different physiological processes. We found actually 2 numbers are specifically involved in the regulating plant immune responses. So we're trying now to establish a cargo spectrum of all different importing betas in plant in order to establish a comprehensive catalog or resource for the community.
Okay, thanks.
Thanks, Joe.
So maybe I ask a question. Yanan, very nice talk. So you mentioned that PNET2 it's a putative chromatin testing factor. This is a very interesting and maybe a bit surprising finding. So my question is a general question. So do you find other new finding related to the nuclear membrane protein or related to nuclear pore complex, you know, the pore complex? So do you find any other new functions?
Yeah, right. As I mentioned, at this moment, CPR5 seems to be a specific membrane component of the nuclear pore complex that involves in regulating the immune activation. We also found another component called PNET1, which is also a membrane nucleoporins in the nuclear pore complex. We think it's regulating the cell cycle process. But that thing is actually very interesting. We think PNET1 is a conserved nuclear membrane component because PNET1 has a homolog in human as well. And in human, that gene is a lung cancer driver. So it expresses very high in lung cancer cells, but if you inhibit its activity, the lung cancer proliferation got inhibited. And if you check their mass spectrometry data, that particular protein actually can interact with multiple nuclear pore complex components as well. So I think those findings are very consistent with ours that this PNET1 is probably a eukaryotic conserved nuclear membrane component that's possibly regulating the cell cycle process. So we're still working on that story at this moment and we've been published. Other than that, actually, we don't know other new members of nuclear pore complex at this moment.
Okay, thank you.
Thanks.
Hey, Yanan.
Hey, Ertal.
Hi. We know I'm quite enjoy your talk. I would like it. Yeah. We know the protein and RNA can pass through the nuclear pore. Can this progress was quickly regulated during in biotic or abiotic stress, just like in cell minutes or?
Yeah. This is actually a very interesting question. So I talked about this GBPL3, which can form the split separation phase and recruit many transcription factors, right? And also the RNA processing machinery. So this enables rapid activation of transcription coupled with RNA processing and transport process. What we are currently working on is whether under different stress conditions, GBPL3 can selectively recruit different transcription factors. For example, under pathogen infection, whether they can selectively recruit many defense-related transcription factors to selectively enhance like certain environmental cue-induced transcription response. At this moment, we don't have evidence to support that, but we are looking at that. And also, I think it's possible, it's very possible that because we have hundreds to thousands of nucleophore complexes in one cell, so these probably can show some heterogeneity, meaning that different nucleophore complexes are probably selectively recruiting different types of transcription factors in order to respond to different types of environmental stresses. So these are all hypotheses that we probably need to test in the future.
Okay, thank you. Sounds very interesting.
Thanks.